Let's see. So we don't have much more to do. I might do a little bit of start with development today. Let's go this way. Right, I'll cut off the exam material after reproductive, because I'm going to get partway through development. I might stick one or two questions on here, but it's basically endocrine reproductive. Certainly heavier on the endocrine. Before I go to the next section, does this stuff work for you? Can you handle the endocrine control of your reproductive system okay? Because here's what, I'm not guaranteeing this is what I'll do, but very often one of the short answers is explain the endocrine control of the female reproductive system. In your answer, describe or discuss, uterine events, ovarian events, and list, the relative concentrations of estrogen, progesterone, FSH, LH, HCG. And that's the question. And you just have to answer. That's kind of a long, short answer, really. But could you do that?
No.
Okay. Well, be able to do it. Now, here's the deal. I may not put that in as a short answer, but if you could answer that as a short answer, any little piece I pull out, you should be able to get, like, for example, what causes ovulation? What is the cause of ovulation?
LH, luteinizing hormone.
That surge of FSH and LH at about the midpoint of the cycle, right? That is the cause of ovulation. What causes menstruation? It is the loss of that functional layer of the endometrium. You would say?
FSH.
No.
Progesterone.
These two target this, right? So the estrogen, progesterone, they're what cause the endometrium to thicken and proliferate.
When they drop.
Levels of estrogen, progesterone drop, endometrium is sloughed off. Those hormones are necessary to support the functional layer of the endometrium. If those hormone levels aren't high enough, you can't maintain endometrium and the cells lyse and sloughed off. So the drop in estrogen, progesterone, that is a direct cause of menstruation. So you will definitely get little bits pulled out. Like what is the function of HCG? Just in general, that one question. What is the purpose of the function of HCG in the human or the female reproductive cycle?
It detects pregnancy.
It does what?
It's how you tell you're pregnant on pregnancy test.
Yeah, but that's not why it's released.
Okay.
That's an artifact. We just use that. Why do you have HCG?
Does it help maintain the corpus luteum?
That's probably the best way to put it. HCG, human chorionic gonadotropin. What does it do? It acts like LH. Remember, LH targets the ruptured follicle and causes the corpus luteum to develop, and it maintains the corpus luteum, but only for about 10 days normally, because LH levels are going to be dropping. HCG is produced when an embryo is implanted in the endometrium. The embryo produces the HCG, but it makes its way to the mother's blood. And HCG does the same thing as LH. The difference is HCG will stay there for months, maintaining that corpus luteum, whereas the luteinizing hormone levels are going to drop a bit. And if there was no HCG, that's, again, why you get menstruation.
So it maintains the corpus.
Maintains the corpus luteum is one way to look at it, yeah. Because you need the corpus luteum. Why? Because that's what's secreting the estrogen progesterone. So if the corpus luteum loses its function, you won't have estrogen or progesterone, which means menstruation will occur, which means no pregnancy, which means extinction of the species. What else? So, just kind of go through it. You can kind of look at it as days. You can think of it and make like a little timeline almost. What's happening on day one? Keep in mind the days are kind of arbitrary. It's a cycle. But day one, all the levels are low. Estrogen, progesterone, follicle stimulating hormone, luteinizing hormone, those are low. Because of that, estrogen, progesterone levels are low, so you have menstruation occurring. That is, the endometrium isn't going to be supported. There are no well-developed follicles in the ovaries. As you go from day one forward, let's say day one, about day 13, FSH, LH levels go up. that causes the follicle to develop, maybe primordial, primary, secondary, mature. As the follicle is growing, that's what's secreting the estrogen, which is then targeting the endometrium. You hit about the midpoint, that's where you get that surge of FSH and LH, follicle ruptures, eggs can make its way to uter2. The continued levels of LH target the corpus luteum, which continues to secrete progesterone as well as some additional estrogen, keep maintaining and proliferating that endometrium. LH levels then drop, about day 14 on, they're going down. As LH levels drop, the corpus luteum regresses, loses its function, estrogen and progesterone levels plummet, and menstruation occurs. So, it's kind of some times. It's really kind of what's happening here, kind of here, and here, and then the cause and effect, and you'll probably be all right. So that is kind of where the test will end. So I'll go over the exam a little bit first, and then I'll start a little bit of development, which will carry over to the next exam. So let's see, this exam will be probably somewhere in the neighborhood of
And when is the exam?
I don't know, when do you want to take the exam?
No.
Monday.
You want to do it Monday?
No.
We can go Monday or Tuesday.
Tuesday.
Doesn't really matter. That means Tuesday, you can ask me any questions, but I'll just go on with development and we'll finish it. And that might be better because Dana's out, I don't know if school, so another day, doesn't much matter. I'll still lecture on Monday, but we won't do the exam until Tuesday. In a way, that's nice. You take the exam, that's all you'll do that day, so you can take it, go, celebrate, cry, whatever it is you need to do after the lecture exam. So probably 45, 50, multiple choice or fill in the blank. And they'll be like the ones on the quiz. So now, hopefully, you know kind of what they'll be like. It'll, probably like, I might put 40 multiple choice down, put 10 fill in the blanks down, something like that. We'll have it on multiple choice. It's about 100 points for those. And then you will get some short answers. Now, what kind of short answers will we get? Well, one I already gave this example like right there. Let me give you some examples of short answers. That doesn't mean I'm going to use them. Doesn't mean I won't use them. I certainly won't use all of them. Let's see. We can do, start with the entrance system. All right. The two examples I've already given you on the quiz. That is a likely short answer question. Just, list all the hormones secreted by whatever. Probably one of the bigger ones, but it's an example on the quiz. List all the hormones secreted by, I don't know, the anterior pituitary or the pituitary. For each hormone, list its action, list the target. So, you can do that as a table with somebody started. That's easy, you don't have to write much. Just the hormones, list them, the target, list them, the action, list it, you'd be good. So that's probably one. Alternative to that, I will just list a bunch of random hormones, and for each hormone, 5, maybe something like that. It'll say for each hormone, list its origin, so you'll need to know where it comes from, list the target, and list its action. So one of those two things, they're similar, they're related. Then, other short answers. I know I've asked in the past, explain how the cyclic AMP second messenger system functions. So you have to go through the steps. Looks like the diagram I have on the PowerPoint. Step 1, hormone binds the receptor. 2, activates the G protein. G protein sends a signal to adenylate cyclase, turning on its enzymatic activity. Adenylate cyclase then converts ATP to cyclic AMP, and cyclic AMP is free to activate protein kinases and enzymes, and you get substrates turned into products. It could be that general. That's a possibility. The other one is mode of action. The difference in the mode of action sort of in general between the lipid soluble hormones versus the non-lipid soluble. There are some questions on it sort of related to that. Lipid soluble diffuses in and out of the blood easily, right? Travels into the target cells, moves directly through the plasma membrane because they're lipid soluble. Binds to receptors in the cytoplasmic nucleus. Usually causes new proteins to be synthesized from scratch. Non-lipid soluble, you have to have increased permeability of the blood vessels around the gland and around the target so that hormone can get in and out of the blood because it can't get across the lipid bilayers. At the target cells, binds to receptors on membrane receptors. Never enters the cell and then initiates some kind of second message. That would be fine. So that's a possibility. What else would that system has? All right, that's a really good question. Maybe do something like, I may say, describe how negative feedback control mechanism works and give me an example. Or describe how a positive feedback mechanism works, just kind of in general, and give me one example. That's a possibility. So 4, 5. Might be part of it, no one's over the house. I often stick one of those in there too. Define terms. So for the endocrine system, there are a bunch of interesting terms. You kind of know what they mean, but they aren't a separate short answer kind of question. Like for example, what is upregulation? What's downregulation? What does it mean if a hormone is? What's a steroid? Hormone making cholesterol. What does it mean? Let's say the difference between what's a bound hormone or a free hormone. Remember we did that half-life. Bound hormones, they're bound to a plasma protein carrier, so they last longer. Free hormones are just naked in the plasma. They have all the action, but they get degraded quicker. So bound and free is a way to affect the half-life of the hormone, how quickly it's removed from the blood. So there'd be a handful of just kind of terms off that lecture sheet, really. What else, Brandon? From blanks, multiple choice, trying to think of Robert's concept, the one I'd ask. And we do a couple reproductive here. All right, for reproductive, again, if I ask you 4 short answers, 3 to probably be endocrine, 1 reproductive, or ask you 5, big 3, 2. So you'll get more on endocrine, because that's the bulk of the material we covered on the exam. But there are a couple potential reproductive system short answers. 1, this one, something about, I can wrap this into a short answer, actually it's kind of a long short answer, but some aspects of the endocrine control of the female reproductive system is one certain possibility. 7. In the male, I can say list the accessory reproductive glands. For each gland, list the secretion and its major contribution to the semen, something like that. So that'd be seminal vesicles, fructose, 60% prostate gland is basic or alkaline, about 30% seems like bulbourethric gland is lubricant, neutralized residual urine, 5%, and the remaining is of course the sperm. So something like that. 8. simple anatomy one I've stuck on there a lot in the past. Again, I give you an example of that, I think last time. Starting at the center of vestibules, list of structures encountered by a sperm as it moves from the center of vestibules, say out the penile region. And you just list them. I'll give you blanks with numbers, so you got them all filled in, you got it all in the right order. That's a possibility.
Like the SG on the board, the order.
And there's a few other more minor ones, it could be, I could ask you to describe some mechanisms or structures that allow you to control the temperature of the testes. I'd be looking for the countercurrent heat exchanger in the testicular arteries and veins, cremaster and dartos muscles that can change the distance. So that would be a small thing, almost fill in blank, really, but that's a possibility. Another just short one, if you look at the uterine layers, we talked about, it's smooth muscle. Why is the uterus smooth muscle? Why are most of your internal organs smooth muscle? That would be something not sensitive to stretch. Again, that's more of a fill in the blank, but it's possible. What else? How do I think of it now? I think of it Monday night. Well, anyway, those are some examples. That's kind of the short answers you might have on there. Might be some of those, might be something new, might be something, but that's the idea. So again, depending on how big they are, if they're shorter ones, you get more. If they're kind of longer, like honestly, if I put that one on, that's a pretty long short answer if you write it out in text, so then you have fewer other short answers. I tend to go with shorter ones, but anything is possible. Can't think of much else. You guys, any questions? Any stuff you might think you might want to look at before the exam Tuesday? Questions about questions?
Are you gonna give us our quizzes back?
Oh yeah, remind me to do that before you get out of here. There's a folder up here on the podium.
Now, please.
Do you ever repeat questions, like questions we have in the quiz you put on the exam?
Yes. I don't always do it on purpose. But yeah, some of those are good questions. I like them. So yeah, you might say, but be careful, because sometimes you see the same question, but it's like, what was that one you had? Which hormones do not bind to, integral membrane proteins, or something like that, right? Well, on the exam, it might say, which hormones bind to integral membrane proteins? So just be careful. You don't say, oh, I remember that one, and put the answer now, because it's changed a little bit, so just be aware of that.
Do we have a quiz Monday?
Pardon?
Like this coming up Monday?
What about this coming up Monday?
Do we have a quiz?
In lecture?
No.
No, you have an exam.
But that's Tuesday.
Well, yeah, it was Monday. No, it's an exam. I will give you a quiz Monday, then one Tuesday, unless you want one.
No. What about in lab?
Lab, you will have a quiz.
Monday?
Monday.
Over hard?
Hard.
Don't worry about it, it's only 5 points. Study for the 100 points.
Right, that's what I say. This one, 100 points. Lab quiz, 80. So, yes, you'll have a lab quiz. Don't sweat the lab quiz much at this point. Worry about the lecture quiz. Way more points. One short answer is worth like the whole lab quiz kind of thing.
So how many short answer will be on the test?
It depends on how long they're in. Usually there's no less than 4, and typically don't go more than 6.
6, it's not bad.
And sometimes I have 10 on there, but I'll put 2 of them together and say, pick one of these, pick one of these, pick one of these. So you have a little bit of flexibility there.
Yeah, it's not bad.
All right. Well. So you kind of got the idea of this. It's going to be like the quiz. Basically, take that quiz and make it 4 times as big. That'll be the format of the exam. You had, 11 or 12 or 13, fill the blank, multiple choice, and then you had 1 short answer. Well, you have about 4 short answers, about 45 of the multiple choice fill the blank. So it's going to be like the quiz, just 4 times bigger. And it's worth 4 times the points. So basically, it's 4 quizzes stapled together. Do a little better than the quiz weren't bad. The last one was a 72 average on the second take, which is decent. Now, somebody would be pissed off with your score. That's all right. Fix it. But they weren't bad. That's a decent average. But averages mean almost nothing when class is small anyway. So just a little bit more, get, the average, the exam average is 72, 73, I will be happy, that's pretty decent. Just saying. So do what you can, you got a few days, you got Friday, you got Saturday, you got Sunday, you got Monday. Stuff out. Email me if you have a question over the weekend. I'll get back to you. All right, the next section we're going to look at, it's usually called embryology. You might see a chapter labeled human development. It's the same thing. We're not looking at a whole lot of it. Development is a little complicated. The terminology is weird. It's one of the harder parts. We're going to just look at kind of some high points, and to me, the parts that are they're easier to conceptualize. You start getting onto some of the weird stuff or just the growth, it gets, a little tedious. So we're just doing some quick, what they call descriptive embryology. This is, based on, many decades ago research where our technology consisted of a microscope, a few bits of, dye and needles and some embryos, and you fertilize an egg, look at it under the microscope and identify parts, maybe map them with some ink so you can see what they developed into later. They just describe what's going on. So, a lot of embryology now is molecular. We can watch all these different parts develop in an embryo. It's really fascinating, it's very predictable. But how the cells know, why do these cells know to be fingers, and not your brain? The signaling that goes on, the molecular signaling that goes on during development, that's where a lot of work is going on now. There's certain chemicals when they're released, they make your fingers form. If you release too much of it, you get extra fingers. If there's too little of it, you get fewer fingers. So a lot of the molecular work is what we're doing in development now. We're not going to mess with that one. It's just kind of some simple, descriptive embryology is what we'll look at. And all I tend to do in this class, I look at very early development, basically from fertilization until the first organ systems are deposited. Basically the nervous system starts to form. Then I stop and I jump ahead and look at those extraembryonic membranes we looked at in lab because they have a lot of interesting environmental or evolutionary concepts associated with them. So we'll look at early development, a little bit of extraembryonic membranes, and that's it. I don't look at growth. For a human, once the development reaches 8 weeks, it's just a human. They call it a fetus at that point. All it does is kind of get bigger and bigger. I don't find that very exciting, so I'll make a lecture on it. So very early development, and then a little bit of later development specific to amniotes of humans. And that's about where we'll leave it. So it's really only about a lecture and a half. Now, embryology typically starts when that sperm fertilizes the egg. That is the first cell for a new individual. It's called the zygote. That's where development starts. But before we get there, I like to go backwards a little bit and look at how you get there. Fertilization, basically. So we kind of looked a little bit at some lab. This is kind of the preamble to actual development. We're not quite there yet, but it's associated. So the first thing we need to look at are the reproductive cells. Of course, we've mentioned the lab. I'll look at the diagrams quickly. This is the male's gamete, or reproductive cell. Of course, this is the sperm. This is an electron micrograph. Electron microscopes give you, much more magnification than we can get in the lab. If you put a sperm under our microscopes in lab, you can barely see them. I mean, they're a little dot, and you kind of see the flagellum, you see a little movement. That's it. These are the smallest cells you'll see in a human. And let me just describe a couple of the characteristics that are important to note. Both the gametes, the sperm and the eggs, are produced by a unique type of cell division, meiosis. Now, just to review briefly, because I know you did this in Anatomy 1, there are two types of cell division, right? You have mitosis and you have meiosis. Mitosis, every cell in your body duplicates itself by mitosis, except for the germ cells, except for the gametes. They're the only cells a human has that reproduce by meiosis. And if you remember, you look at human chromosomes, of course, that's where the genes are, the genetic material. The chromosome number for humans is 46, right? Arranged in 23 pairs, remember that? Every cell in your body except your sperm and eggs, and the name for that is a somatic cell. If someone refers to a somatic cell, what that means, that is every cell in your body except your oocytes or your sperm. So every cell in the body except the reproductive cells are called somatic cells, and they are referred to as being diploid. You remember the term diploid? Nod your head because it's been a while. All that means is you have the chromosomes arranged as 23 pairs. So every cell in your body, except your sperm and eggs, has 46 chromosomes arranged as 23 pairs. They are produced by mitosis. If you remember what mitosis does, if you have a cell with 46 chromosomes, it reproduces by mitosis. What do you end up with in the daughter cells? 46 chromosomes, right? Mitosis is just cloning. Mitosis, you're making identical copies of identical cells just over multiple generations. Nothing changes barring, random mutations or something. That's how you produce every cell in your body, except the sperm and eggs. Now, sperm and eggs are produced by meiosis. That was a little different. You remember what the end results of meiosis were? Again, you start with one of your cells with 46 chromosomes, call that the parent cell. What was the difference between mitosis versus meiosis? You remember? Just anything, really.
They were all genetically distinct at the end.
All right. A couple things about meiosis. One, it happens twice. They'll have what they call meiosis. One, the parent cell divides into two, and then each of those resulting daughter cells will divide again, so you end up with 4 daughter cells, whereas in mitosis, you only have 2. That's a difference, but then genetically, that's what's important. All of your cells have 46 chromosomes, but if you look at your sperm or your eggs genetically, they only have half the normal chromosome number. They're referred to as being, the term is haploid. Because they're produced by meiosis, they only have half the chromosome number. Basically, you separate those 23 pairs. So you don't have 23 pairs anymore, you just got 23. You lost, you don't have them. That's meiosis, that's what you went over last semester. And yeah, each of these is genetically distinct because of several factors, the random assortment of the homologous chromosomes and crossing over. There's a number of ways we get these to be genetically distinct. It's like a male, if you think about it. Let's say a male has, 10 kids. It's the same male, same mom, same dad, they have 10 kids. So genetically, they're the same parents. Why don't all the kids look the same? Shouldn't they? Same 2 parents, reproduce with their genetic material, produce this offspring. Reproduce again, produce this one. Do all the kids look the same? No, they look similar, but not the same. Again, that's because in meiosis, in a human, there's millions and millions of possible ways to separate the chromosomes. So basically, no 2 sperm are the same, no 2 eggs are the same. So there's a little genetic variation there. They just put together different combinations of chromosomes, basically. But that's meiosis. That's what you looked at last semester, and they'll refer to it as a nuclear event. If you look at the nucleus, you have these 46 chromosomes. Daughter cells have 23. All right, that's meiosis. In spermatogenesis, it is still meiosis. The germ lines in those seminiferous tubules, those cells undergo meiotic division, so they'll ultimately divide twice, and you do produce 4 daughter cells with 23 chromosomes. But then, as you're actually producing the sperm, again what we call spermatogenesis, the actual formation of the sperm, genetically it's meiosis, but one of the reasons sperm are so small, as they're being formed, they shed most of their cytoplasm. So that's why these sperm are the smallest cells in a human body. As they undergo meiosis, genetically, they also shed almost all the cytoplasm. So what you have with the sperm, very tiny cell, and it's got these 3 parts I've already mentioned, the head of the sperm here, and all you'll find in here, it's got a big nucleus. So sperm's, it's almost like a nucleus on a stick. There's one other organelle that's going to be important for us here. It's called the acrosome. I'll mention it in a bit. That's important for fertilization. Here's that little mid piece or body that has mitochondria in it. I'll just draw one out, right? And then you got the flagellum. Of course, these are mobile and they leave the male's reproductive system. They swim towards an egg. But there's not much in there. You're going to shed most of the cytoplasm. That's partly why you need fructose from the seminal vesicles as an energy source. There's nothing stored in the sperm. Like I said, it's basically a nucleus stuck on a flagellum. There's not much else there. So they're very tiny. Now contrast this sperm, the oocyte. Now this is a diagram because I don't have a good picture of a human egg. I'll show you some micrographs of human eggs in a bit. This blue thing here represents the egg. Now, genetically, the sperm and the egg are equivalent. That is, a sperm has 23 chromosomes. Eggs are also made by meiosis, in the ovaries, and they will have 23 chromosomes when they finish meiosis. But the difference, while sperm are some of the smallest cells, eggs are huge. I mean, a human egg isn't that big, but like I mentioned before, you can see it with your naked eye. If you look at those ovary slides closely at the light, you'll see those primordial follicles, which are basically the oocyte. You can't see the squamous cells around them. And if you look at chicken egg, that yolk in the middle of a chicken egg, that's the egg. Technically, that's one cell. So eggs are gigantic. They are produced by meiosis, but why are they so big and the sperm so small? Well, during what's called oogenesis, which is actual formation of the eggs, genetically it is meiosis, but remember meiosis, mitosis as well, they're nuclear events. They don't even talk about what's happening in the cytoplasm. So what happens when the germ cell of the eggs divides, that first division in meiosis, the cytokinesis is unequal. What you end up producing after that first division, you have two daughter cells here. They both have 23 chromosomes. They're genetically equivalent. However, one of the daughter cells gets all the cytoplasm, the other one gets none. This thing is called a polar body. It's genetically equivalent to the egg, but it can't do anything. It can't grow, can't divide. Even if we're fertilized, nothing happens. There's no raw materials, nothing in it. And then, of course, these cells will go through meiosis too, and the same thing happens. You got one giant cell, and the polar body may also divide. So at the end of oogenesis, or meiosis in egg cells, you end up with 4 daughter cells, 1, 2, 3, 4, but 3 of these that are formed are polar bodies. They're non-functional. They'll just disintegrate, they can't do anything. That's partly why the oocyte is so big. One of the daughter cells receives all of the cytoplasm, so it's huge. So these egg cells are pretty big. So that is the oocyte. Rest of the results of meiosis just degenerate. And this is kind of giving you an idea of the size scale. This thing in blue, that's the oocyte. These little things are representing sperm. They're much, much tinier. Now, of course, for fertilization to occur, all that has to happen, the sperm's plasma membrane, cell membrane, has to come in contact with the egg's plasma membrane. When that happens, basically the sperm nucleus is going to be injected into the egg. That is all the male contributes. The rest of the sperm degenerates. So you get everything from your mother except 23 chromosomes. That's all dad contributes. Some centrioles in some species, but it's basically just the nucleus. So here's where we'll talk about this a little bit of a problem here. In order for a sperm to contact the egg, there are some layers in the way. That's what this slide is sort of showing you. This is the egg's plasma membrane here. The sperm around here. So if this were a human, this would be occurring in about the first third of the year 2. That's where fertilization usually occurs. How is this sperm membrane going to contact that egg cell membrane? You've got all this other junk on the outside. Junk is not a technical term. That's what we use for now. Well, what is this junk? All right. Right outside the egg's plasma membrane, which is blue, do you see this yellow zone here? That's a layer of, it's protein and water. It's a sort of protein mixture. It has a lot of water tied up with it. It's called the zona pellucida. This is extracellular. It's like this layer or this case the egg cell secretes outside of it. So this zona pellucida, think of it as a protein layer outside of the egg's plasma membrane. So it's secreted by the egg. Then out here, this is cellular. These things are representing cells. You know what this layer is? I mentioned it in lab. What is that red layer on that diagram representing? Anybody? Think about what we looked at in lab, because we actually looked at this on a micrograph. Yes, there's some speculation. All right. In the ovarian follicle, there were a few parts of that you have to identify, right? You got the oocyte, you got that fluid-filled antrum. You remember there was a ring of cells around the oocyte? We called it the corona radiata. That's what this is. When the egg is ovulated, when that follicle ruptures, The egg, the oocyte is released, but it still has the corona radiata ringing it. That's what this is. These are granulosa cells. These are maternal cells. And they're calling them cumulus cells here, but that is the same thing we refer to as a corona radiata. These are maternal cells stuck on the outside. So before a sperm can fertilize that egg, it's got to get through that layer of granulosa cells. It's got to get through the zona pellucida, and only then can it contact the ectoplasmic membrane and fertilization occur. Now, I have a few diagrams on this PowerPoint that are not in your textbook at all, and I cheated a little bit. Your textbook doesn't have very good pictures for most of this, honestly. I see that in the most recent edition. But what you have to keep in mind, most of embryology, it is not, certainly the early stuff, was not done on humans or in fact any placental mammals. Because imagine, how easy is it to study the development of an embryo in a placental mammal? And that could be a dog, a deer, a human, a rat. It's technically difficult. Why? Because where does the embryo develop? In the uterus. You can't take it out, put it in a petri dish and study it, it dies. So up until fairly recently, and now we can go, you didn't get a lot of work done on placental mammals. They were too hard to use, technically too difficult. So much of your early embryology work, all of it, was not done with placental mammals. We used other species as models. The most common ones in early embryological work were sea urchins, starfish, a group called echinoderms. Now, they don't look like us at all. You don't see starfish with those 5 arms. Evolutionarily, they're actually fairly closely related to chordates, to the group humans are in. So their early development is actually somewhat similar to ours. And the reason they use them, because I use these in another lab where it's got a 4-hour lab, it's principles of biology. But we do an in vitro fertilization lab. We use sea urchins. You take a starfish or a sea urchin, you shoot it up with potassium chloride, which of course would kill a person. Those guys don't have a nervous system to speak of, so it doesn't bother them. But you shoot them up with potassium chloride. If it's a male, it spews out sperm. If it's an egg, female, it spews out eggs. They have external fertilization. So all you have to do is get a microscope slide, put some eggs on it, drop some sperm on it. You can watch all this early development. So this group called echinoderms is a very common model for early development. The other ones that were commonly used and still are, frogs. Again, why frogs? Well, you take a male, you can shoot the males and females up with hormone treatments, and you can make them breed whenever you want. It's external fertilization. Male spews or female spews out the eggs, male the sperm, you can put them right on a microscope slide or a petri dish, watch fertilization, watch early development. It's easy. The other group, which is actually very similar to humans, are chickens, birds. Why? Because again, you can't really see fertilization, but you can get the egg, crack it open, and you can watch it develop. And birds develop just like humans. I mean, if you look at an early chicken embryo, early human embryo, you can't tell the difference really unless they look the same. But we don't use placental mammals. So what's happened as the years go by, you had this group of biologists working on sea urchins and frogs, and they named all the parts they saw. Then later, when we had the technology, we started looking at placental mammals. It was a different group of people in different labs. Guess what they did? They sort of named things different. So the only reason I'm telling you that is so I don't confuse you. This is not a human. Some of my slides are actually frogs or sea urchin eggs, and the reason for that is I have better slides. The structures are similar, they're not identical. But what you see here, egg plasma membrane, in the diagram, you got the zona pellucida, and you have this outer layer here. Notice this green layer. In a human, that was the zona pellucida. You'll notice it has a different label here. You can ignore it in this class. In anything but a placental mammal, they call it the vitamin envelope. In the placental mammals, it's zona pellucida. It's the same thing. So ignore that stuff like that. I mean, for our purposes, this is going to be functionally the zona pellucida. The other thing that's different, you've seen frog eggs, right? What's on the outside of frog eggs you see sitting in the pods and puddles that go out back to one? It's not a clump of cells, is it? They're covered with jelly. It's a little different because they release their eggs differently. Outside of these eggs, this is functionally the same as the corona radiata, but that's not what it is. It's acellular gelatin that they basically deposit on the eggs as they're released. It's going to have the same impact on fertilization. I just wanted to mention that because, we're not looking at frog eggs in here, and I'm just telling you, you're not responsible for anything that has, specifically to do with frog eggs or serous eggs. I'm gonna use them in a lot of examples, because again, you have more information, it's easier to get the details. Experimentation with placental mammals and embryos is difficult, and human embryos, it's nearly impossible, because there's all kinds of ethical issues in there as well, so we use frogs and serous eggs instead. But this is basically the same thing. So here's what we got. This is the outside, so if this were a human egg, this would be the fluid in the uterine tube, let's say. Here's your outside here with its extra layers. Now, of course, what's gonna happen, the sperm is gonna fertilize the egg. But here's a couple of questions that are kind of interesting. If I took some sperm, freshly ejaculated sperm, plopped it in a petri dish, and plopped in an egg, a human egg, would they fertilize? Probably not. It's kind of weird. Sperm, in order to fertilize an egg, they refer to the term they have to be capacitated. It turns out sperm that are initially ejaculated can't fertilize an egg yet. They actually have to swim through the female reproductive tract before they have the capability to fertilize the egg. That's kind of weird. And I think what happens is, do you remember the role of cholesterol in cell membranes? Cell membranes are mostly phospholipids, right? And proteins, with carbohydrates. There's cholesterol in animal cell membranes. The cholesterol makes the membrane harder. Well, when sperm are first released, they have a lot of cholesterol in their membrane. It's a fairly rigid membrane. As they swim through the female reproductive tract, the cholesterol is basically washed out of them. So by the time they reach, that last third, the first third of the uterine tube, they're capacitated. They've lost enough of the cholesterol that the membranes are fairly fragile, and when they contact the egg cell membrane, they can open. So that's just kind of interesting. The other thing I want to mention, if I took one sperm, plopped it in a petri dish, and I took one egg and plopped it in the petri dish, assuming the sperm was capacitated, chemically treated, it's ready to go, would one sperm fertilize one egg? What do you think? Yeah? So, probably not, because think about it. In human ejaculate, there's 400 or 500 million sperm, and there's only one egg ovulated. Man, that's a really low efficiency. Why so many sperm? If it only took one, ejaculate 2 or 3, save the rest for later or something. Why do we ejaculate so many sperm? It's because one won't do it. If you took one egg and one sperm and put them together, here's what happens. This sperm, here it is, swim along. They are attracted to the egg, and there's some, what do they call it, positively chemotactic. They can tell the chemicals released in ovulation, and they'll swim down the uterine tube, and they'll eventually encounter the egg. Now, when they do, what does the sperm do? Well, remember, here's where this acrosome is going to come in. Remember, a sperm, the head of the sperm, has a nucleus with the DNA, but there's also a small membrane-bound organelle at the sort of tip of the head of the sperm, this thing right here on the right end. We call it an acrosome. The acrosome contains, well, they sometimes call them acrosomal enzymes, that's not very descriptive. They're hydrolytic enzymes. They're enzymes packaged in this acrosome, which is really just a vesicle, a membrane-bound compartment. And they are digestive enzymes, they're hydrolytic enzymes. They will tear up organic molecules. So what normally happens when this sperm contacts the outer layers of the egg, they will undergo something called acrosomal reaction. It's kind of what they're showing right here. What happens is the membrane kind of releases, you rupture that acrosome, all of these enzymes come out of the acrosome, and what they're going to do, they're kind of indicating here, they erode these outer layers. So the acrosomal enzymes will degrade, the corona radiata cells. They'll digest through it. They'll go through the zona pellucida, they'll digest through it. So the acrosomal enzymes, their function, why they're necessary, is this allows the sperm to basically clear away these unnecessary outer layers so that it can reach the egg's plasma membrane. Now here's why, one of the reasons why you need more than one sperm. Sperm are teeny, they're small cells. If I have this winny little sperm here, it's going to release a few of these enzymes, but enzymes don't last forever. They're going to get neutralized by enzymes floating around in the fluid. They're going to diffuse away from the site. So if one sperm is all you had, it would come in here, carry out an acrosomal reaction, digest away a little bit of this stuff, but then that's it. It would run out of enzymes, lose function, get washed out, get neutralized, you're done. You haven't got to the membrane yet. This is partly why it takes large numbers of sperm. So there are going to be thousands of them out here. And they're all slowly, conducting these acrosomal reactions and digesting their way through the outer layers of the cell until finally one of them reaches the egg's plasma membrane. And of course, it's not 400 million, you do release 400 million in ejaculation, but some of those sperm die in the female reproductive tract, some of them go through the wrong uterine tube, some of them get chemically confused, start trying to fertilize, epithelial cells or something. But even by the time you reach where the oocyte is, there's still tens of thousands of those sperm, so it does take a large number. So sort of collectively, they'll erode through these outer layers, and eventually one sperm will make contact with the egg plasma membrane. And what they're showing you here, this red blob here, that's representing the sperm's 23 chromosomes. They're going to be injected into the egg. So at that point, you had 23 chromosomes in the oocyte, you got 23 in sperm. That's it. You're back to the 43 or diploid number of chromosomes. That's a new individual genetic. But there's a couple of weird things that happen here that I just got to mention because it's weird. Weird, well, interesting. All right, here's the experiment. What you're going to do is you're going to take a petri dish, and you're going to take a human egg that's viable, plop it in the petri dish. Then you're going to get hold of some hamster sperm, and you dump that hamster sperm in with that egg, and not put them to fertilize. Tens of thousands of hamster sperm. Is that hamster sperm going to fertilize that human egg? And then you make like Hamster Boy, when you go to the checkout line, you see him in those magazines next to the checkout, right? Can you do that? Would it work? Because people have tried it somewhere and sometime. Would it work? No. Well, why not? I mean, it seems creepy, hamster boy, that's not going to work. Why would it work? All right, it doesn't. Here's why. It's kind of weird, but it's an interesting evolutionary mechanism. And it may help to look at a non-human species, because we do have internal fertilization, and you probably should be aware of whose sperm gets into your reproductive tract under those conditions. But most other species don't have internal fertilization. For example, look at sea urchins, look at clams, look at corals, oysters. A lot of those marine species, do they actually copulate, have internal fertilization? No, something like an oyster is just stuck on a rock. Here's the male, female's over there. Sorry, honey, I just can't make it. Because they don't move, right? Coral doesn't move. They use just broadcast spawning, basically. At a particular time of year, it's usually, keyed in on springtime. The males dump out all their sperm, the females dump out all their eggs. They're just in the oceans, mixing around that way. It works fine. But the potential problem is during something like spring tide, many of the species on a coral reef will all try reproducing at the same time. And if you ever see videos of it on the Discovery Channel, it looks like soup, man. The clams are releasing sperm, the corals are releasing sperm, the echinoderms are releasing sperm, the worms are releasing sperm, they're all releasing eggs and sperm. We have this egg sperm soup. I mean, it's everywhere. And so you might have a sea urchin's egg, and it could get fertilized by a clam sperm or something like that. This would ruin the reproductive output of the individual. I mean, it takes a lot of energy to make eggs, right? They are, at least in an energetic sense, it's expensive to produce eggs. And if some other species, like if you're a sea urchin and a clam fertilizes your egg, the embryo's not going to develop. They're not genetically compatible. So all those eggs that get fertilized by a different sperm, you'd be wasting them. So it's inefficient. So what's been produced is a mechanism of what's called species recognition, or at least close to species recognition. What they're showing on this sperm, there are some little proteins. They weren't very creative with the names. They're calling them binding proteins. And again, proteins are genetically determined, right? So if you're a particular species, your sperm are going to have a particular type of binding that is recognizable as only that species makes it. It's genetically unique. On the egg, kind of projecting through the egg's plasma membrane, these little red things are showing here, those are binding receptors. So it's almost like a hormone receptor, binding, or an enzyme substrate binding. The binding proteins and the binding receptors have to be compatible, which basically means they have to be from the same species, or at least really closely allied species. So if this were hamster sperm, it would get to this point, and if those binding proteins can't bind to that binding receptor, no fertilization. But if it's the right species sperm, the binding proteins will bind to the receptors, and then the sperm can release its genetic material into the egg. Now, what is kind of interesting, if you take a human egg and you strip off the zona pellucida, you can chemically do that. When you strip it off, you also get rid of the binding receptors. So if you take a treated human egg, put it in a petri dish, dump in hamster sperm, guess what happens? Then you will get fertilization. Do you get a hamster boy? No, we're not genetically compatible. The embryo won't even develop, but fertilization will occur. So there is species recognition between the eggs and the sperm. They have to be from the same species, or at least closely allied species. If not, you can't get fertilization because there's no molecular recognition. So let's say we got the right species of sperm in here, binds to the egg, they all undergo this acrosomal reaction. You erode these outer layers. Eventually, one sperm makes it in, boom, dumps in its 23 chromosomes. So we got 23 chromosomes from the sperm. The egg had its original 23 chromosomes. That is fertilization. We're good to go there, but there is a potential problem here. Any idea what it might be? How many sperm are still out here? There may still be 50,000 sperm. They're still conducting an acrosomal reaction. What would happen if another sperm contacted that egg cell membrane and dumped in 23 more chromosomes? That embryo wouldn't develop, at least not very far. They call these polyploid individuals. Haploid is one set of chromosomes. That's how sperm and eggs. Diploid is every other cell in your body. If you got another 23 chromosomes in here, the embryo typically isn't viable, not in humans. Because think about it, in a human, if you get like one extra chromosome, I think it's 21, that causes Down syndrome. People with Down syndrome don't have 46 chromosomes, they got 47. Now one extra, they're not missing anything, but that one extra chromosome interferes with development enough that it causes some significant damage. If you got another whole 23 extra chromosomes, that embryo won't even develop, because it won't develop very far. It might divide a couple times, that's it. You have to let one sperm genetic material in, and then it's got to stop. If you get more than one sperm fertilizing the egg, again, you've wasted your reproductive effort. So if more than one sperm fertilizes the egg, I have a name for it, it's called polyspermy. You must prevent polyspermy, or the embryo will not be functional. And it turns out there's a couple mechanisms that do that. In humans, really only have one. There's a couple mechanisms that actually fix you. Again, most of this early work was done on other species, not humans. I'm going to stick a slide up. Don't take notes on it. You don't need to know it. Your textbook used to have this, 15 years ago, because again, we assume what we find in other species probably applies to humans. But then we get to the point where we can do the detailed work to find out the detail, and we find, oh, that doesn't work. What happens in most species, this is a depolarization. You remember that from like the nervous system? You start at -70 for an action potential, then you go up to positive 30, and then back to that. This is a depolarization. This is the membrane potential in a sperm, in an egg, before it's fertilized. As soon as that sperm contacts the egg, there's an electrical change, just like in a nerve cell. So if you took an egg, as soon as that sperm enters, if you were measuring electrically, the inside of the egg goes from -70 to, +5 or something. That's a depolarization. That change in voltage actually temporarily repels all the sperm. Now, they call this fast block to polyspermy. We thought it occurred in humans until about, 20 years ago. Then they actually took human eggs and measured them and go, oh crap, it doesn't happen in humans. So we don't have this. So the reason I say that is all we have is slow block to polyspermy, and they don't mention anything about fast block, which is weird. Slow block to polyspermy is what humans have. Now what happens in our eggs, I'm gonna draw myself a picture over here. So let's say this is the egg cell membrane, it's kind of big, and then we'll have a few more colors in here. So my red is kind of representing the zona pellucida, kind of this blue line here. The black is the cell membrane or plasma membrane of the egg. And then you might have a little, corona radii out here, or jelly coat, depending on the species. All right, so let's say we get a sperm, comes in here. I'll use blue. Digests its way through, releases its 23 chromosomes. Right, that's what is happening, right here. We got this sperm in the egg at the 23 chromosomes. Now keep in mind, there's many more sperm trying to get in here. We don't want any of them in here, or the embryo will not develop. We'll waste our reproductive efforts. the first thing that happens, there is a calcium flux in human egg cells. It's not associated with an electrical change, but what happens is when that first sperm contacts the egg, it opens calcium gates. Calcium diffuses in, and it acts as a signal. So there is a bit of an influx of calcium, but we don't have the electrical changes we saw in most other species. Then the other part to notice here, these things right here, we're calling these things cortical granules. All right. All these are, if you look at them under a microscope, they look like little dots just underneath the membrane of the exoplasmic membrane. They're basically vesicles, so you can call them cortical granules or cortical vesicles. These are little membrane-bound packets that contain some kind of stuff. Now here's what happens. When that first cell contacts the egg cell membrane, the calcium coming in makes these vesicles migrate toward the exoplasmic membrane, much like a synaptic vesicle migrates toward a presynaptic cell. And then it will merge with the exoplasmic membrane. So let's say I've got some right here. Merge with the exoplasmic membrane, and the contents that were inside the vesicle are going to be released right here. Now this space is kind of the space between the zona pellucida and the exoplasmic membrane. If you look at this diagram, do you see these little brown bars they're showing right here and here and here and here? I drew as red lines. There are proteins that actually connect the zona pellucida and the exoplasmic membrane. They are physically connected together. So what you're seeing here is the egg cell membrane, zona pellucida, they're physically connected. There's proteins tying them together. Well, it turns out the secretions from these cortical vesicles, they degrade or digest those proteins. So what that's doing, you are now breaking the connection between the exoplasmic membrane and the zona pellucida. Those proteins are digested. Then the secretions from these vesicles are hypertonic. They're highly concentrated compared to the extracellular fluid. And what that's going to do, what they're indicating here, it's going to cause water to move in by osmosis. So the contents of these cortical vesicles, they're released in the space between the exoplasmic membrane and the zona pellucida. They enzymatically digest all the proteins holding those two layers together. Then they cause the osmotic influx of water. And what they're trying to show you here is, look what's happening. As the water moves in, it's filling this space up right here. And what that does, the osmotic pressure basically pushes the zona pellucida away from the exoplasmic membrane. Now the reason that helps is there's still a bunch of sperm out here. So as you push that zona pellucida away because of the osmotic pressure, you're taking all the sperm that are still out there and you're pushing them further away from the egg. You're increasing the distance. It makes it more likely they'll fertilize right away. So you're increasing the distance between the plasma membrane and the zona pellucida, carrying any sperm away with it. And then finally, the chemicals in these cortical vesicles, they chemically alter the zona pellucida. They chemically tan it or harden it. And it becomes impervious to the sperm's acrosomal enzymes. And they change its name. So what was the zona pellucida, it detaches from the plasma membrane, gets pushed out because of osmotic influx of water, and then it gets chemically altered into a fertilization membrane. It's chemically altered such that it doesn't matter how many sperm are out there now, this thing is chemically resistant to the acrosomal enzymes. Sperm can be there all day long, it doesn't matter, their enzyme will not get through it ever again. This is a fertilization membrane. Here, since we're pretty close out of time, let me show you a couple of pictures. Try to remember to bring out videos next time. Like I said, I do this early development and fertilization in a different class, we use Searton eggs, and I've got some videos of it, but this is a picture. You can see the moment these things are fertilized. What you're seeing here, it's a Searton egg. This is the ectoplasmic membrane right here. The scale, these little black flecks you see out here, those are sperm. But as soon as one egg fertilizes, or one sperm fertilizes this egg, the zona pellucida pushes out. You can see it. See this clear band out here on all of these? That was a zona pellucida. And you can just watch it. As soon as that egg, one sperm fertilizes the egg, you'll see this membrane up one side and push out all around the egg. That is a fertilization membrane. So just looking at these have been fertilized. How can you tell? If it wasn't fertilized, you would not see that zona pellucida out here. It would be physically attached to the ectoplasmic membrane, which makes it not visible. You can't resolve it. But as soon as it's fertilized, you see this membrane just bulge out. And I'll mention this a couple more times because remember, we talked about it in the lab, cleavage, there is no growth of the embryo, right? So this, what we have inside this membrane here, this is a zygote, that's first cell. It's going to divide many times. I think I have one more picture out here somewhere. Let me show you what I mean. We'll talk about cleavage next time. But this looks like about an 8-cell embryo. It's getting close to a morula. So you see now the cells are tiny, but you see this clear thing out here? That's still a fertilization envelope. A fertilization envelope is rigid. This is one way you know the thing doesn't increase in size. You start out with one cell in this big envelope. The cell divides, the envelope stays the same size. That's it. Now in a human, let's switch to another video here, another picture I mean. Here's what's happening in a human. Here is your oocyte being released. It's moving down to uterine tubes. It's fertilized right here. So that's what will be about the first third of the uterine tube. This is typically where the sperm will swim up. And as we'll see, the early development, we'll talk about next time, that cleavage, that's occurring as the thing's moving through the uterine tube. But this white thing around it, that's the fertilization envelope. It's not getting any bigger. And in fact, when we talked about tubal pregnancies last time, what prevents a tubal pregnancy is that fertilization membrane. As long as it's in there, it won't implant. What we'll see, when you go from the morula to the blastocyst stage, you see it right here, they say hatching out of the fertilization membrane. That's when the embryo grows, it sheds or rips out of this fertilization membrane. That's when it can implant. As long as that fertilization membrane is intact, it won't implant. That's typically why it doesn't implant in the uterine tube, why you normally don't get a tubal pregnancy. Anyway, that's the fertilization membrane. And that process of separating the zona pellucida from the plasma membrane, pushing it out osmotically, and then hardening it chemically, that's referred to as slow block to polyspermy, because I think it takes like, I don't know, 20 seconds. We don't have a comparable fast block because it just apparently doesn't happen in humans. So the reason they call it slow block, there is a fast block, you just don't see it in humans. Fast block is a depolarization, it takes fractions of a second. This does take 20, 30 seconds. And usually, that's going to prevent any additional sperm from getting into the egg. It doesn't always. Like on those slides I showed you, those Searton eggs, I pack in so many sperm on some of those, they do get polyspermy embryos, and they develop all funny, and they never make it to the larval stage, because we overload this system. But normally, with the number of sperm normally present, that slow block, it occurs, and you get no more fertilization, and then hopefully development will continue as it should. That is it for fertilization. And that's probably where I'll stop today. We're a little early, but now what we'll do next, now is when development starts. So we got that sperm in the egg, that new cell now, the new 46 chromosomes, if it's a human, that's the zygote. So development starts now of the new individual. We start with that first cell, the zygote, and we'll go through until we get to an embryo with a couple organ systems forming. That's not where we'll stop. And we'll quit for today. And the other words go, man, how weird words in development. They're hard to say. There's gastrulas, gastrulation, blastulas, I don't know. It's just, the terminology is a little weird. There won't be that much of it, but yeah, some of the words there, that always seems weird to me. I don't think they want them. But we'll finish up with this, because development, one more lecture, we're done. So we'll do development on Monday. And if you have any questions for the exam while you're studying over the weekend, bring them in. I don't have a problem going over anything again, over endocrine or anything else on the exam. That's fine. You can do whatever you want. But, if we don't have any questions on the exam, and I don't know if we'll have an hour, 15 minutes worth, I'll finish up development. And the next thing on the syllabus is Mendelian genetics, but I intended to skip it this semester. I've skipped the last 2 semesters because of time, and still in the same weeks, I'm not sure how well we're doing with time. Mendelian genetics has some really interesting stuff in it. The reason I can justify skipping it, you've all done it probably since junior high school. You know little Punnett squares, where you take the alleles, you do the, that's all it is, and most people have done it multiple times, so I think, and I'll probably say it this semester, I may just give you a smart book on genetics, and ignore it, and then move on to the next organ system, which will be lipid development, and that would be the circulatory system. And that is mostly what exam 2 will be. It'll be this little bit we go development, a couple of questions, we'll be done with development in the next lecture. Then it's almost all circulatory system. We're gonna go circulatory system for 8, 9, 10 lectures. Takes a while, hopefully it makes good sense, because part of the reason I spent so much time in the circulatory system, if someone goes to the hospital, there's something wrong with them around you don't know what it is, you have to guess, what is it likely to be? The circulatory system, we get hypertension, we have a heart attack. We have a lot of issues with the circulatory system, it's a common, I don't call it a weak spot, but it's an interesting system that affects all the others, so I spend a lot of time with circulatory. That's basically what the next game is, circulatory and development. Having a fun and exciting weekend, but not too fun and exciting because we have an exam early next week. Yeah, did that thing record the whole time? That's awesome, it did. Sometimes it shuts down, low battery or what? It sure did, look at that.